Hey everyone, Professor Davis here, and now we're going to investigate the principles of disease and do an introduction into what we call epidemiology. So before we get started, we do need to look at some definitions, and we're going to look at what we call pathology, infection, and disease. So pathology, guys, is the study of disease. So when you end up taking like a pathophysiology, it's the study of disease and how it affects the function of those body systems. So pathology is the study of disease. What does that include? Well, first, it's going to include what we call the etiology. Etiology is the cause. What's causing the disease? What's causing the illness? Then it's going to look at what we call pathogenesis. Pathogenesis is how the disease develops. How does it start? What does it look like in the middle? And what does it look like at the end? Okay, so pathogenesis. The next one is going to look at the effects of the disease. So how does it affect the structures of the body? How does it affect the functions of the body? And so that's going to be the part of the effects. Now, a little bit different definition is infection. Infection is when a pathogen invades you. So it's the invasion of the pathogen, okay? The disease, however, is the physiological effect, how your body reacts to that infection. That's the disease. So you need to be able to distinguish those. So example here, an HIV infection may be asymptomatic. You don't even have any symptoms. But after a while, the effects of that HIV will then progress to the disease of what we call AIDS. Okay, so that is just an example. All right, so hosts and microbial relationships. So on our body and in our body, we have some normal flora. This normal flora is also known as your normal microbiota. So you have bacteria living on you and in you, which is completely normal. These are known as your permanent microbial residents. Okay, so they live there, they should be there, not a problem. They a lot of times will help prevent invasion of different pathogens, the ones who make you sick. This is known in biology as competitive exclusion principles, meaning two things cannot compete for the same resources within an ecosystem. So this is why lions and tigers don't live in the same place. They're both big cats, they're both predators. They would not be able to survive together because they would hurt each other. The same thing can happen here. Because you already have bacteria living on you, it can hurt other bacteria so that way you don't get sick. Factors such as antibiotic therapy, though, can lead to a disruption of the dynamics of your normal microbiota. This happens a lot of times when you take antibiotics and you end up getting gut infections or things like C. diff because the normal microbiota also gets disrupted with the antibiotics. And so it allows these pathogens the opportunity to make you sick. And these guys are known as being opportunistic because if they get the opportunity, they will do it. However, there are times you have some microbial agents that are transient. They're the ones who don't live on you normally. They're temporary microbial residents, and these guys typically do not cause disease. All right, so here is an example of some normal flora, also known as your normal microbiota. So these are all areas that open to the outside, as well as certain organs that are going to be key places for bacteria. So you can see in the nose, the throat, the large intestine, the mouth, the skin, the vagina, and the urethra. These all have different types of bacteria present. Okay, or other microbes like yeast and things like that. So what do these body areas all have in common? Well, they all involve cutaneous or what we call mucosal or mucous membranes. Okay, cutaneous means that it's like the skin structure, it's on towards the outside, and the mucosal is obviously the ones who make mucus. What do most of these organisms have in common? So if you'll look behind each of the organism, you'll see a lot of them have the G+, okay, and then they'll have a C for coccus, B for bacillus. You'll see some have a G-, and you see some that have yeast. If you'll notice, the majority of these are gram positive. So many of these are going to be gram-positive microbes. All right, this leads us to the idea that if you're a host, this means that you're having a symbiotic relationship with those microbes. Well, what is symbiosis? Symbiosis is when two or more organisms are living together. And there are three types of symbiosis. There is commensalism, mutualism, and parasitism. So let's look at each of these. The first one is commensalism. With commensalism, guys, one of the organisms benefits, but the other is neither benefited nor harmed. Okay, so this would be a bacteria who lives on you and you provide it with like food and shelter, but it doesn't help you and it doesn't harm you. That would be commensalism. So we provide the nutrients and space for it to reside, but we get no benefit, but we're also not harmed. Okay, that's commensalism. The next one is mutualism. Mutualism is where both organisms benefit. So both the host and the other organism get something good. So example, we provide nutrients and space for the microbe to reside, but then they help us in the digestion and providing us vitamins that we normally wouldn't get. All right, and so we both are benefiting from this. The last one is parasitism. Parasitism is when one organism benefits, it's the parasite, but the other is harmed. That's known as the host. So for us, this would be if a microorganism is now harming you, it's causing you to get sick or have pain, then that's a parasitic relationship. What type of microorganisms are going to be involved in parasitism? These are going to be things like viruses, pathogenic bacteria, some types of protists, and some of our helminths. If you'll remember, those are the worms. Okay, so they would participate in parasitism. Now, pathogenic, only when a special set of circumstances occurs, that's going to be when they are opportunistic. So note, when typical normal microbiota that is either in a mutualistic or commensalistic relationship, if they get the opportunity to make you sick and become a parasite, sometimes they do. Okay, so like staph, you have staph on your skin right now. But if you get a cut and that staph gets in there, you could end up developing a staph infection. That means that staph went from being commensalistic to parasitism. So it depends. Now this means that these guys are what we call opportunistic pathogens. They're normally harmless organisms, but they can become harmful. Well, how do they become harmful? Well, one, they might be relocated. If they're relocated to a different area than where they actually help, they could become harmful. E. coli is an example of this. E. coli, when it's in your intestines, is great and it helps you. It gets you vitamins. It helps you finish digesting your last little bit of food. It's wonderful. The problem is when it gets somewhere else, it causes an infection. So this example is that it causes urinary tract infections or UTIs. The next way that they can become harmful is if there's a break in the skin or the mucous membrane. The barrier that normally keeps them out of your body gets broken, so they get the opportunity to get into your body. This means your first line of your body's defense has been compromised. The last one is when the host itself, you yourself, are compromised. This is when you have an immune system issue, specifically immunosuppressed patients. These can be patients who don't have an immune system like we see with AIDS patients. This could be a patient who is undergoing lots of chemotherapy, so their immune system is hindered and altered. This could be somebody who's on anti-rejection medications because they've had a heart transplant, so their immune system has been suppressed. So there's a lot of different types of immunosuppressed patients, okay? And that can cause some of our bacteria to become opportunistic. All right, so now let's talk about the etiology of infectious diseases. Now remember, etiology is the cause, what's causing it. An infectious disease is a disease that's caused by a pathogen that is transmitted either from person to person, okay, so you got it from somebody else, Or you got it through a vector. This vector could be a mosquito, it could be a tick, it could be a fly, but something that probably bit you. Or it could be acquired by your environment. You were exposed to it because it was on the door handle or it was in the air or something like that. Okay, so acquired by the environmental exposure. Now this brings us to a scientist that we want to take a look at, who is Koch. We talked a little bit about him at the beginning of the semester in one of our early chapters in Unit 1. However, we do want to look at Koch's scientific research a little closer. His research was designed to establish a correlation, a connection between a pathogenic microbe and a disease. So this is the idea of cause and effect. The cause was the microbe, the effect was the disease. This is what he worked on doing. So how did he do it? Well, we're going to look at his experiment, but we do need to note that his experiment did have limitations. Some things that we now know, but we were able to find out based on his research. So these limitations include things like not all organisms can be easily cultured. Okay, some examples of these are going to be the causative agent of syphilis, okay, Trypanosoma pallidum. This one cannot be cultured on a normal media like we do other types of bacteria. Another is the Mycobacterium leprae, which causes leprosy. It cannot be cultured. And then obligate intracellular parasites like rickettsia. These guys have a hard time. Now, why is this a limitation? Well, let's take a look at his experiment. So if you look at this picture right here, you're going to see that first off, he found that the mouse died, okay? So he wanted to figure out what might have caused the death. And so what he does is he takes a sample, he looks at it underneath the microscope, and he decides to culture it. So if you take a look here, you see that the mouse dies. We're going to see what caused it to die. So he took some of the fluids and he cultured it, and he grew some bacteria in colonies. He then took those colonies and he looked at them underneath the microscope to try to determine what they were. Okay, this idea is if they're coccus or bacillus, are they in a strand, are they in clusters, that kind of thing. Okay. He then took what he cultured, he put it in a syringe, and he injected it into a new mouse. If this mouse died, he then recultured their fluids, and he compared to see if what killed the first mouse is also what killed the second mouse. And again, this is great as long as things can be cultured on the media. These guys can't. All right. Another limitation is that some infectious diseases like pneumonia and meningitis have poorly defined etiologies, meaning it's hard for us to see the progression in how those diseases are going to manifest, what those causes are. And the reason is there are many different causes for those diseases. You can have fungal pneumonia, you could have viral pneumonia, you could have bacterial pneumonia. Okay, same thing with meningitis. You can have the same things for meningitis. So it makes it hard to know exactly what is causing it. And that's a limitation here. Another limitation is that some pathogens cause a variety of disease. So on the last slide, we saw that a disease could be caused by multiple pathogens. Here, one pathogen can cause multiple diseases. Okay, so the pathogen that they picked here is Streptococcus pyogenes. Streptococcus pyogenes can cause scarlet fever. It can also cause cellulitis. It could also cause erysipelas, rheumatic fever, post-streptococcal glomerular nephritis, and necrotizing fasciitis, which is flesh-eating bacteria. So one bacteria can cause all these different diseases. Well, now we know this and we can study it a little bit better, but these were limitations to Koch's experiment, even though he gave us a lot of knowledge when he developed this experiment at the beginning. So let's look at classification of infectious diseases. The first thing we need to look at are what we call signs and symptoms. So guys, when we see symptoms, symptoms are subjective, meaning they depend on the patient. So this would be a lot of times if a patient is saying that they have a certain pain level, or they say they've been throwing up. If you can't observe that they've thrown up, it's considered a symptom because it's what they're telling you. Okay, you can't test their pain level. All right, as much as we would like to, we can't. All right. But the other one is what we call the signs. Signs are measurable and they are objective. So this would be like their heart rate, their blood pressure, if they have a fever. Those would be signs. So I have a little quiz here on this particular page, and it's asking you if it's a sign or a symptom. So when the patient says they have lethargy or fatigue, well, that would be a symptom. What about if we take their temperature? Temperature is going to be a sign because we can measure their temperature. What about anxiety? We are determining it based on what the patient tells us, so it is a symptom. Tachycardia, very fast heart rate. Well, we can measure that, so it's a sign. Pain, we use the pain scale. We tell them, hey, what's your pain from 0 to 10? But that's still going to be a symptom. It's subjective. What about a rash? You can see a rash. You can observe a rash. And so because of that, that's going to be objective. So that's a sign. And then numbness. Numbness, they're telling you it's numb. You don't know for sure. You don't know if how they're feeling it. And so because of that, it's going to be a symptom. All right, so these are just some examples. And the last one on here is hypertension. Hypertension is high blood pressure. And again, because we can measure that, it is a sign. So let's talk about diseases that are transmittable from host to host, okay? So if a disease can travel from host to host, we normally explain it or characterize it as it having a high spreadability. So it can spread very easily. So there are some terms we need to take a look at. The first here is if the disease is what we call communicable. Communicable means it can spread from person to person, okay? A communicable disease can also be contagious. Contagious is going to mean that it's easily passed to somebody else. These are things like the flu, tuberculosis, and measles. They can easily be passed to another individual. However, when we look at this idea of communicable and contagious, these are going to be our infectious diseases. They can cause infections. On the other hand, the non-communicable, the ones that don't pass really easily from person to person, this is going to be food poisoning, tetanus, and a type of mycoses reaction. This is dimorphic mycoses, which is a fungal issue. So these guys are going to not be passed from person to person. You can't give food poisoning to somebody else if you've had it. It's non-communicable. Okay. The next thing we want to look at is called occurrence. Occurrence is going to look at two main things. The first is the incidence. The incidence is the number within the population that has the disease within a given period of time. Okay? So it's the number of people who have the disease for a given period of time. The other term we want to look at is called prevalence. Prevalence is the number within the population that has or has had the disease within a given period of time. So this, you'll see the prevalence is bigger because it's everybody from past and present. Whereas incidence is right now. Who has it right now? Okay? And so incidence and prevalence are a little bit different. The next one is frequency. Frequency is going to relate to how often it's seen. So if it's sporadic, we only occasionally see the disease. And you can see that in the diagram here in B. If it's endemic diseases are constantly present in particular populations. So they're going to be found in particular areas and they're predictable. We can actually predict when we're going to see those diseases. We then have an epidemic. Epidemic is shown here in picture C. This is where the disease is statistically seen at a higher rate than expected. So even though we might have expected the disease to be there, there's a higher rate of it that we did not expect. That's known as an epidemic. And then we can go a step further where we call it a pandemic. A pandemic is an outbreak of the disease on a worldwide basis. And again, we lived through one of these in 2020 where a disease went and it caused havoc worldwide. This is not the first pandemic. We've had many throughout history, but this is one of the most recent ones when we talk about COVID. The next kind of terminology we want to talk about is severity or duration. Okay, how severe is the infection or the disease, and how long is it going to last? If it's an acute infection or disease, it's going to have a sudden onset. It comes on quickly, but it also is short-lived and goes away quickly. You can see that here in the red. This would be an example of influenza, which is the flu. Chronic diseases are going to develop gradually. They're going to slowly develop, but they're going to be continuous for a long period of time. They are going to be persistent. They hang around. And they could maybe reoccur. And if they reoccur, that means they come back. A really good example of this one is mononucleosis, which is mono for short. The last one are the latent. Latent infections or diseases become active after being inactive. So this would be the virus such as shingles or the herpes simplex. So shingles is actually caused by the chickenpox virus that you might have been exposed to and you had chickenpox, but then it sits latent and dormant and it could reemerge, but it doesn't reemerge as chickenpox, it reemerges as shingles. That is known as a latent reaction or a latent disease. The next one we want to look at is the extent of the infection. The extent of the infection is where it's kind of located. So does it have a local area? It's restricted and it's in a small area. It's more controlled, like an upper respiratory infection or a lower respiratory infection, but it's controlled in that area. Is it systemic, meaning it affects the whole body? So it's generalized through the whole body. This a lot of times can occur if the cardiovascular or lymphatic systems are involved because the infection can move from one place to another using the blood or the lymphatic fluid. Okay, now this could lead to a very focal infection where infection is going to be more localized, but it originated from a systemic infection. All right, this brings us to a few more terms. When we're looking at some of these terms, we have what we call septicemia. Septicemia is when organisms like bacteria, viruses, or fungi, as well as their toxins, get into the blood. Septicemia is also a lot of times termed as blood poisoning. The next one is bacteremia. Bacteremia is where bacteria is found in the blood. This can actually occur just from you brushing your teeth. You have a lot of bacteria in your mouth and the chemistry of your mouth, and as you brush your teeth, if you swallow some of those contents, that bacteria could enter your blood. We then have toxemia. Toxemia is when toxins are in the blood, and viremia is when viruses are in the blood. Okay, so we can get very specific on what we're looking for in the blood. Now, sometimes sepsis and septicemia are used interchangeably. However, they're not really the same thing. Sepsis is your body's inflammatory or immune response due to the infection, and sepsis could lead to your organs failing and cause you to go into septic shock. Now, normally sepsis happens because of septicemia, but it's the reaction of your immune system that causes the sepsis. All right, the last kind of set of terms we want to look at here is going to be primary versus secondary. Guys, a primary infection or disease is the initial infection or disease. Okay, so HIV and pneumocytosis, which are a bacterial infection, followed by a viral infection. Okay. Secondary infections are those that usually are from being opportunistic. So if you've ever gone to the doctor and they said, well, you have the flu, but you also have strep throat. Okay, you probably got the flu first. That was your primary, your initial, and then you developed strep throat because it was a secondary opportunistic infection. Okay, and that's what we see happening here a lot of times. So, what are some predisposing factors that lead us to having these types of infections? Well, for UTIs, one of the big ones is gender. Okay, you're going to see UTIs found more in females than males. And there's a reason. The urethra is a lot shorter in females, so bacteria has an easier chance of getting up into the bladder and the kidneys. We see in males it's a lot harder to do this. Also, the fact that the openings are a lot closer to other openings like the vagina and the anus. And so transfer of bacteria or yeast can happen very easily here. Another predisposing factor is stress. A lot of times stress is going to cause us to develop a type of infection, especially an opportunistic infection, because stress causes your immune system to be depressed. Another one is nutrition. Nutrition is important because you've got to make sure that you're eating properly. If you're not giving your body the nutrients it needs, then it can't fight invaders very well. And then, of course, the season. You'll know that we have what we call seasonal allergies. We also have a flu season. Those are going to be times during the year when those things are more prevalent. Okay, so like the flu is more prevalent from November to April. It doesn't mean you can't get the flu in June. It just means it's less likely to happen. So the next thing we want to look at is what we call the development of disease. How does disease develop? And we have a chart here that shows you how a disease goes up and causes all of our symptoms and problems, and then how it decreases after your immune system takes care of it. And we're going to look at these piece by piece. So the first section of this diagram is the incubation period. The incubation period is the time between exposure and your actual symptoms. Okay, so symptoms haven't appeared yet, but you've been exposed, but you don't have the symptom of, say, like a fever or a headache or anything like that. Okay, so this is called the incubation period. The next phase is called the prodromal phase, or the period. This is the onset of mild signs and symptoms. So this is when you start having the runny nose or the cough or the low grade fever. Okay, these are going to be the mild signs and symptoms. Then we have the period of illness. The period of illness is the most severe signs and symptoms. So this is when you normally feel the worst. Okay, and so this is seen here in part C. It's shown in the actual kind of pink structure. It's the most severe. Then we move on to the period of decline. The period of decline is where you start to recover. Your signs and symptoms start to go away. Now, they don't go away completely, not yet, but they're starting to subside. They are decreasing. You are starting to feel better. And this brings us to the last period, which is called the period of convalescence. The period of convalescence is where you return to your pre-disease state, but you have to be careful here because a lot of times you can still be a carrier of the disease and you could pass it on to other people. You just aren't sick anymore. Okay, so this is someone who is still infectious but has no symptoms. Okay, so this is somebody who is still infectious but has no symptoms. All right, so let's take a look here. Below is an overview of what is needed in order for an infection to occur. First, there must be a reservoir that harbors the pathogen. So if you look here in this diagram, you see that the first area shown here is the reservoir. What is harboring the pathogen? Is it a human? Is it an animal? Does it transmit from animals to humans? Is it non-living? So like, is it located in the soil, the water, the food, the air? Where is its reservoir? Okay. Second, if you follow the arrow, we have that there must be a means by which the organism is transmitted to the host. So how did it get from the animal to the human? How did it get from the soil to the human? How did it get from human A to human B? That's called transmission. There is contact transmission that can be direct, indirect, or through droplets. We also have a vehicle, meaning it comes from water, food, or the air. And then we have vectors like arthropods, which are bugs who could transport the infection. And then the third thing that has to happen is the host must be susceptible to the disease. Now, just because it got transmitted to you doesn't mean you'll get sick. It just means that you are more likely because you've been exposed. Your susceptibility determines if you're going to be sick. So can it gain entry into your body, like through your membrane, your skin, or through what we call a parenteral route? Can it attach? If it can attach, can it penetrate and evade your cell's defenses? There's a lot of things that have to happen in order for you to get the infection. Okay, so let's take a little closer at the spread of infection. So with the spread of infection, guys, we see that the reservoir is the source, and it can either be human, animals, or non-living like soil, water, food, or a fomite, which means like a surface, an inanimate object, like a desk, a door handle, things like that. All right, so when we talk about a fomite, it's any inanimate object, and they could be used to transmit infectious diseases. So let's look at a reservoir that's the zoonosis reservoir, the ones that come from animals. So if you take a look here at this particular chart, it shows you some viral infections, some bacterial, fungal, protozoan, and helminth, and it shows you where the reservoir is. So if you look at influenza, some types are going to come from swine and birds. Okay, if you look at rabies, those are bats, skunks, foxes, dogs, and raccoons. So it's showing you the reservoir of where they come from. And in this particular case, they all come from a reservoir of animals. But they can be transmitted to us as humans. Okay. Another one that we see here is the mode of transmission. How does it travel from, say, the animal to us, or the air to us, or the water to us? Well, the first is going to be from direct or from contact. The first here is direct contact. Direct contact transmission is from person to person. It's not going to require an intermediate object. This is due to touching and kissing. These are going to be one of the main things where transmission is happening. Indirect contact transmission is going to be there's an agent that transfers it from a non-living object. Examples would be that you get this from a towel, eating utensils like sharing a fork or a spoon, thermometers if they're not cleaned properly, stethoscopes, bedding, clothes, money, syringes, a door handle, a desk. These are all examples of fomites that are going to be where the organism gets transferred to us. The last type of contact is droplet transmission. These are microbes that are spread in mucus droplets that can travel a short distance, so less than a meter. And guys, a meter is about 3 feet. Okay. Examples of this are sneezing, coughing, even talking can release this, as well as laughing. Okay, so this is your contact transmission. The next transmission is what we call vehicle transmission. Vehicle transmission is where it's transmitted by water, food, or air. And you can see these in the examples. So water is if you drink water that's contaminated, like from a stream or a pond or something like that. Food could be where you don't prepare it properly. We talk about specifically chicken can carry salmonella. And if you don't cook it all the way through and you don't clean off your surfaces after you cook the chicken, we do see that it could pass on a disease, be a vehicle. And then the air. If you look here in the air, they're blowing up a lot of the contents and it could cause illness. Another example with the vehicle, though, could be bodily fluids. So if it's transmitted through blood, urine, semen, things like that. The next one is the vectors of the arthropods. These are the ones like insects, mites, ticks, mosquitoes, flies, and other bugs that transmit the disease to the individual. They carry the pathogen and they move it from host to host. Now, there's two ways they can do this. They can mechanically do this by getting on their bodies. So this would be like when the fly lands on the trash is going to potentially have some sort of bacteria present. Then the fly lands on your sandwich and then you eat your sandwich. You didn't eat the fly, but because it transmitted the bacteria over, it could make you sick. On the other hand, we do see that the vector could be biological, where they actually bite you. And when they bite you, they inject the infectious agent, the bacteria, the virus, the protozoa, whatever it might be. Some examples of this are going to be things like plague, malaria, yellow fever, typhus fever, Rocky Mountain spotted fever, Chagas disease, and Lyme disease. And that's not all of them, but that's quite a few. Okay, so let's take a look at some of these vectors a little bit closer. So if you take a look here, it shows you the disease. It tells you what the pathogen or parasite is. And then this red box shows you the vectors. You'll notice a lot of these vectors are going to be things like mosquitoes, sandflies. There's also some black flies located in here, or even ticks. But these are all different things that will bite you. Okay, so they bite you to transmit the disease. Okay, so we talked about the reservoir. We talked about transmission. Now let's talk about the susceptibility of the host. So how susceptible is the host to the disease? Well, are they really young or are they really old? Because guys, when you're really young, your immune system is not developed yet, so you're more likely to get sick. And if you're really old, your immune system is tired and you're also more likely to get sick. Another thing is are you immunocompromised? Do you have HIV or AIDS? Do you take medications that inhibit your immune system? Do you have lots of stress in your life? These would all be examples that could cause immunocompromised issues. So guys, if you look at this, this is a chain of transmission. The reservoir leads to the portal of exit, which then goes to the mode of transmission, the portal of entry, the susceptibility of the host, the infectious agent, and then it starts over again at the reservoir. Since this is a chain, we need to break the chain to prevent the spread of infection. Well, where are we going to normally break the chain? Well, you're going to break it either at the reservoir or you're going to break it at the mode of transmission. Okay? So these are some of the areas where we work on breaking that chain of infection. All right, this brings us to healthcare associated infections, or HAIs. These were formerly known as nosocomial infections, but now we call them healthcare associated infections. These are microorganisms that are prevalent in hospital environments or like in nursing homes. These guys are going to ultimately affect people who are compromised. Now, if you're in the hospital, you're compromised for some reason. So this increases your risk. The chain of transmission is normally direct and indirect contact are the most common. So we have to work on breaking those. So how do we control these healthcare infections or these nosocomial infections? Well, you have to break the chain of transmission. So this means if you're working in the hospital or you're seeing a patient, whether you're visiting or whatever, you need to wash your hands. Wash your hands before you go into the patient's room. Wash your hands after you leave. Another thing is hospitals a lot of times need to use what we call expendables, where we only use them one time. So if they get contaminated, they're destroyed instead of trying to reuse them for somebody else. We also see proper disinfection is important. So knowing what kind of disinfectants we can use in the hospital that are good at killing the different infections that are present. You also need to make sure that we sterilize the equipment. This is true when we talk about the equipment for like surgeries and stuff like that. And sometimes we have to use what we call reverse isolation. Reverse isolation is where the patient is put in isolation for their protection so that we are not potentially bringing in a bunch of diseases to them. A lot of times when somebody is in isolation, if you're going to go visit them, you have to put on a gown, gloves, a mask, and it's to help protect the patient. Now, hospitals do have infection control committees that they use. They monitor infections on a regular basis to see if they ramp up. And if they do, they try to figure out what's causing that. And sometimes it's a carrier, somebody who is carrying that disease and they don't even know it and they're passing it from patient to patient. So some common microbes that are associated with healthcare types of infections you can see here, like the Coagulase negative staphylococci, this is the most common cause of sepsis, and we do see that a lot of it is antibiotic resistant. You can also see like Clostridium difficile, that's C. diff. It causes nearly half of our hospital acquired or healthcare acquired types of diarrhea. So there's a lot of different ones here, and this is just a few. The next thing we want to look at are the sites of infection. Where do most of these infections take place? If you'll notice, we have this pie chart here, and it shows you the most abundant is the urinary tract infections at 32%. Okay, so these are infections we're talking about in healthcare areas. We also see surgical site infections are pretty large at 22%. Lower respiratory infections make up 15%. Bloodstream infections make up 14%, and other is your 17%. This just means that it accounts for all the other ones that are not part of the ones listed. All right, so this just shows you that different sites of infection and what we can maybe do to stop the transmission. The last little thing we want to talk about here in this particular chapter is what we call epidemiology. Epidemiology is the study of when, where, and how a disease is transmitted. This is going to include looking at the incidence rates and also the frequency, how often you see it. Epidemiologists are normally concerned with how diseases are spreading. Okay, so they're not concerned on like how to treat them. They're just concerned on how they're spreading and how they can stop that. This is going to require a lot of case reporting. So certain diseases are required by law to be reported to the CDC. The CDC then takes that and then they use it to try to establish or break the chain of transmission. The CDC stands for the Center of Disease Control. This is not the only one. This is just the main one here in the United States. We also have the National Institute of Health, which is the NIH. And you have the World Health Organization, which is also known as WHO. Okay. Now, the CDC does have certain diseases that are nationally notifiable. And when we notify the CDC about the disease, we also have to include the morbidity and the mortality rates. And these are normally weekly reports that hospitals send to them. So they report how many incidents they have. That's the morbidity. They will also report the mortality, which is the number of deaths. So some emerging infectious diseases are those that are new or changing diseases that are increasing, or they have potential to increase in incidence. So some examples of, say, some bacteria ones are going to be things like MRSA, VRSA, C. diff, and E. coli. Now you can notice here that the year of emergence goes back to 1975 on this chart, and it goes and runs until 2001. This chart could be expanded. There are a lot of other ones, but these are ones who were emerging during those times. Another chart here shows you the viruses. These include things like SARS, H1N1. H1N1 is a type of flu virus. Okay, that flu virus is going to be one that was very, very big. It was a big issue, and it reemerged back in 2008 and had some problems. You also have avian flu, West Nile, Ebola, HIV. We also have a new virus on here, what's called Zika virus. We have some other viruses too, like COVID-19. Purons, remember those are the proteins that misfold. This is going to be Creutzfeldt-Jacobs disease, or CJD. So these are just some examples here that show you, okay? So how do we report this stuff? Well, we're going to report it to the CDC, and with that in mind, the CDC gives guidelines on how to protect against those. And one way that we can protect against some of these diseases, not all of them, but some, is the recommended scheduled vaccinations. So here's an example. This shows you a list of all the different types of reportable diseases that can be sent to the CDC based on if we see them in the hospital. And a lot of these, when we talk about viruses, have vaccinations that are recommended to help take care of them so that they don't become as big of a problem. In order to do this, 80% of the population needs to be vaccinated. This creates what we call herd immunity. Because guys, not everybody can be vaccinated. Some people are allergic to vaccines. Some people don't vaccinate due to religious reasons. But if 80% of the population is vaccinated, it can help them. So this is a list. These are the ones that are the scheduled vaccinations that we have a lot of proof that they help protect against these diseases reemerging. This is things like diphtheria, tetanus, and pertussis. This is known as the DTaP shot. This is a shot that you do get boosters on occasionally, and if you work in the medical field, you'll get more boosters. We also have the measles, mumps, and rubella shots. We saw there was an outbreak recently in an area around us in Texas that we saw measles again, and we had some deaths from it. It's a childhood disease that is completely preventable. If you take the vaccine against measles, you're protected 98% of the time. That's a really good protection. So measles, mumps, and rubella. We also have some hepatitis here, hepatitis A, B, and then also poliomyelitis, which causes polio. And then you have the Haemophilus influenza type B. Vaccinations are available for some others, but they're not necessarily routine vaccinations. The doctor may recommend them, but they're not necessarily as effective as some of these are. Another thing to note is some of these diseases that we report to the CDC are foodborne, meaning that you get them from contaminated food. Some examples are things like botulism, E. coli, listeriosis, salmonellosis, and shigellosis. These are all going to cause types of food poisoning. You may have seen this in the news where like the peanut butter got recalled because it was contaminated with E. coli. Lettuce, certain canned goods, all those things get tested. And if we start to see a trend where they all came from the same kind of chicken company or they all came from the same brand of peanut butter, we need to take a closer look and we may need to take it off the shelf. Some of these on the list are also sexually transmitted. This means they're transmitted due to sexual contact. These are things like HIV and ultimately becomes AIDS. We have the cankeroid, chlamydia, which is genital here, gonorrhea, and syphilis. Now you'll notice that herpes is not on this list because guys, we're going to see later that majority of people in the United States have a type of herpes. Doesn't mean you have genital herpes. It just means that you probably have the herpes that causes cold sores in your mouth. So we can't really track that very well. On this list as well, you have some that are transmitted through vectors. These are like Lyme disease, plague, Rocky Mountain spotted fever, and yellow fever. And yes, the plague is still around. Here in New Mexico, we actually saw a death recently with somebody who got the plague. Plague is transmitted through fleas. Okay, Lyme disease and Rocky Mountain spotted fever, those are transmitted through ticks. And then yellow fever is normally through mosquitoes. The last one I want to show you here are those that are going to be transmitted through animals, the zoonoses. These are going to be things like anthrax, brucellosis, plague, ehrlichiosis, hantavirus, psittacosis, Rocky Mountain spotted fever, and rabies. These all are going to be transmitted from animals to humans. Okay, and so some of these are listed on here. You'll notice some we didn't really talk about, but we will talk about a lot of these when we get to each of the chapters about disease. So when we talk about the diseases of the respiratory system or the digestive system or the urinary system, a lot of them are going to come off this list that are going to be diseases that are reported to the CDC. All right, so this finishes up this particular look at the intro to principles of disease as well as epidemiology. So if you have any questions, please let me know.